This figure is showing the process of mitosis broken into 4 specific stages. And the names of these stages give an idea or relate to where the chromosomes are during the process of mitosis. Now, we need to take just a moment and go back and review the purpose of mitosis. Mitosis is a form of cell division that results in the production of diploid cells. Now, what's a diploid cell? Di, the prefix means two. Ploid refers to sets of chromosomes. So a diploid cell is one that has 2 sets of chromosomes. You get one from mom and one from dad. So in humans, the diploid chromosome number is equal to 46. We have 23 pair of homologous chromosomes. We get one member of each pair from mom, the other member of each pair from dad. So if we think about what mitosis does, it results in the production of diploid cells, or the phrase is used in the reading 2N cells, because 2N is the symbolism used to represent diploid. So in thinking about it in more detail, if 2N is equal to 46 in humans, then each daughter cell that results from mitosis must also have 46 chromosomes in it. So what does that mean the chromosomes in the original cell about to undergo mitosis need to do before division takes place? And the answer is DNA replication. And so you'll remember in our previous discussions, we said when the DNA of a chromosome replicates, it produces sister chromatids. So prior to mitosis in human cells, there will be 46 chromosomes lined up end to end. One chromatid from each chromosome will go opposite directions, so that after the cell divides, which is called cytokinesis, after division of the cytoplasm, you will have 2 daughter cells, and each daughter cell will have 1 unduplicated chromosome from each pair, and that results in the production of a diploid cell, or again, a 2N cell. So let's take a look at this particular figure, and this also gives us some ideas or some information about what's called the cell cycle. So before the cell undergoes division, it's in what's called interphase. Interphase used to be called a resting phase of the cell cycle, but actually that's a misnomer. During interphase, transcription, translation are taking place, DNA duplication is taking place. In other words, the cell's replicating things to get ready to divide. As the cell enters into the first phase, which is prophase, the nuclear membrane starts to break apart and the chromosomes start to condense. So you can see we have 4 chromosomes in this cell, each chromosome containing 2 sister chromatids. So we would have 2 homologous pairs of chromosomes, the big ones and the small ones. Now, one member of each pair comes from mom, the other comes from dad. In metaphase, the replicated chromosomes line up end to end along the cell. In anaphase, the sister chromatids separate, and 1 chromatid from each chromosome goes each direction. So you'll see we have 4 chromosomes going this way, 4 chromosomes going this way. Telophase marks the end, or the last phase of mitosis, and this is where the separated chromatids start to decondense. In other words, they start to unravel, so they are in preparation to enter into the next phase, which is interphase, and the DNA will be used for duplication, transcription, translation. Now, coupled with telophase, which is the last of the stages of mitosis, we have cytokinesis, which is the actual division of the cytoplasm. So one cell goes through nuclear division, which is prophase, metaphase, anaphase, and telophase. Nuclear division is followed by cytoplasmic division, which is called cytokinesis, and it results in the production of 2 daughter cells. Again, each of these daughter cells would be 2N. So in this particular hypothetical example, 2N is equal to what? And your answer should be 4, because there are 4 chromosomes. Each daughter cell will have 4 chromosomes, but they will be unduplicated chromosomes. So a 2N cell results in the production of 2N cells. Each one of these daughter cells is diploid, or 2N.